Good morning, welcome to Science News. We're here in New Orleans at the 2013 Fall Conference of the Council for High Blood Pressure Research, the American Heart Association. I'm Greg Fink, I am chair of the association, chair of the council I should say, and we're here today to talk with Dr. Justin Grobe, who is one of our finalists for the Goldblatt Award, a very important award of our council that recognizes an early independent investigator for their newly independent contributions to science. And Goldblatt awardees typically go on to be important researchers in the area of hypertension and leaders of our council. So that's another reason why this is a particularly important award. So Justin, I want to start off by asking you about your own personal background in terms of your educational and research background. In other words, so how did you get to where you are now?
Sure. So I grew up in Mason, Michigan, graduated high school there and went on to college at Hope College in Holland, Michigan.
Excellent.
I graduated with degrees in biology and chemistry. Then I went on to the University of Florida and worked in pharmacodynamics and received my PhD there. After graduating with my PhD, I went on to the University of Iowa for postdoctoral training in Kurt Sigmund's lab. And after about 6 or 7 years of being a postdoc, I decided it was time to join the faculty. So I was very excited to be accepted onto the faculty at the University of Iowa. So that's where I am now.
And you're an assistant professor there?
Correct. I'm on the tenure track assistant professor in the Department of Pharmacology.
Oh, good. Excellent. So the other issue that, of course, we like to talk about with our finalists is the work that got them here, the science that you talk about. You talked about yesterday at the meeting. Could you give us a capsule summary of the work?
I'll try to.
Okay, great.
So my research interest in particular is focused on the crosstalk between metabolic control systems physiology and cardiovascular control systems physiology. In particular, my work is focused really on the brain renin-angiotensin system. The RAS is well known as a circulating hormone system, and many of our antihypertensive drugs target the renin-angiotensin system. But in fact, the RAS exists in many tissues where it can act as a paracrine or autocrine or even intracrine system. There's a lot of evidence that angiotensin is used as a neurotransmitter. And so a lot of our work is focused on understanding how that may contribute to both cardiovascular and metabolic control. In particular, my work that I described in the Goldblatt competition is investigations into ways in which the brain renin-angiotensin system contributes to metabolic control on behalf of other systems. In particular, we focused on the requirement of the brain renin-angiotensin system for leptin to mediate some of its metabolic rate and cardiovascular effects. And so a lot of this work really implicates the brain RAS as an integral part of metabolic and cardiovascular control.
And so leptin is the hormone that's produced by the adipose tissue or the fat cells, right? And it signals the brain. What does leptin do when it acts on the brain again?
So leptin induces a lot of different things, especially with regard to metabolic physiology. It is an appetite suppressant, and it also stimulates metabolic rate, and is suggested that it stimulates blood pressure, which leads us to some clinical implications. We believe that leptin, which is grossly elevated during obesity, we believe that that contributes to the hypertension that occurs in many patients that have obesity. There's obesity-associated hypertension. And we believe that the hyperactivity of the brain renin-angiotensin system that results is probably causative for hypertension in that case. So,
tell me a little bit about the model you use. I know you use some very interesting, creative models to answer your questions. Tell me a little bit about that.
Sure. So, in particular, my training as a postdoctoral fellow in Kurt Sigmund's lab was to really learn how to develop and utilize novel transgenic and knockout animals. And so the particular animal that we've used for this study, we've deleted the angiotensin AT1A receptor in all cells that express the leptin receptor. And this had a very potent effect to completely ablate the metabolic rate stimulatory effects of leptin, which is pretty amazing. And doing the math, it worked out that over 2 weeks of a high-fat diet, which induces leptin, we saw that the animals failed to increase their resting metabolic rate at all, as opposed to wild-type control littermate mice. It stimulated it quite a bit. So,
do you think there's a possibility that abnormalities of the brain renin-angiotensin system contribute to human obesity? I mean, obviously it's a very, very important problem now in the U.S. and around the world. Is this a potential source of that problem?
So we really do believe that the brain renin-angiotensin system is hyperactive and probably causative for hypertension in a broad spectrum of hypertensive disorders. Of course, in humans, it's rather difficult to assess brain renin-angiotensin
Yes, of
system
course.
activity.
Of course.
So it's difficult to study directly in humans, but in virtually every animal model of hypertension, inhibition of the brain renin-angiotensin system can ablate the hypertension.
Right.
So pretty strong evidence there, and we think that, you know, if the shoe fits for 99 of the problems, then probably it'll fit for the last one too. So
That makes sense. You know, so a question that comes up a lot when we hear about studies using transgenics in animals is whether or not this technology is going to be ever transferable to humans. I mean, clearly some of these targets are druggable targets as well, but what's your, I'm interested to ask for your personal view now, whether you think genetic-based therapy, gene therapies will ever be sort of a mainstream approach to treating some of these chronic conditions.
So as a pharmacologist, the biggest power that I believe that genetic targeting in animal models can tell us is it really can inform us in terms of pharmacogenetics and pharmacogenomics. Identify the patients who need different types of therapies. Hypertension in particular is a very wide disorder.
Yeah.
It's many different diseases added together to one symptom.
Yeah.
And identifying patients who will respond well to different types of drugs is one way in which genetics will inform us. Also, when I worked at the University of Florida with Mike Katovich and Mohan Razada and Colin Sumners, they of course use a lot of viral vectors to manipulate the genetic makeup of an animal. And some clinical trials have been done with viral vectors and genetic targeting, but it's the regulatory problems and of course just the fear that everyone has of using such a powerful tool when we don't really have, you know, a lot of sort of honing in on the control of it yet. Something 5 years from now, probably not. Something 50 years from now, it wouldn't surprise me.
Excellent. I really appreciate your insight in this as an expert, because it's something that people, you know, hear a lot about and are really quite interested in. Justin, I appreciate you taking the time to come and talk with us today, and I would just tell everyone out there listening to the Science News today or watching this video that if you'd like to be able to come and experience this kind of science and the kind of information, very up-to-date information that appears at this meeting, please come and join us next year at the meeting in person. It's going to be in San Francisco in September, so please do join us. And again, thank you very much for being with us this morning.